Recombinant DNA Technology.
Definition of recombinant DNA technology is a method in which DNA molecule made in vitro with segments from the different sources or by combining genes from different organisms.
Explanation for this definition is naturally DNA recombination occurs randomly during meiosis 1 through crossing over of homologous chromosomes during fertilization when two gametes fuse or through mutation or transformation.
Recombinant DNA technology also involves modifying and recombining DNA to produce desired products such as animal or plants with desirable traits.
By using DNA technology, DNA recombinant can be manipulated to serve specific purpose.
For example, the mass production of human insulin by bacteria. In order for the bacteria to produce human insulin, its genes must be modified.
Hi, I am Izati. So right now I will talk about target DNA. Do you know target DNA is the first tool of recombinant DNA technology? Okay, so target DNA is the sequence of DNA containing gene of interest. For example, is gene encode. Gene encode for protein which can dissolve blood clots. Gene encode also known as gene therapy. Gene encode used in heart attack therapy.
Assalamualaikum, I am Nur Akila. The second tools of recombinant DNA technology, which as you can see is restriction enzyme. Okay, during an infection, restriction enzyme which cut and cleave a DNA molecule at restriction site. This restriction site is the short base pair sequence that contains 4 to 8 base pair. This restriction enzyme which cut and cleave a DNA molecule at restriction site to produce a short restriction fragment. A different restriction enzyme cleave at different base sequence. Okay, on restriction site there is also contains palindromic. Okay, palindromics is the same sequence of bases is found in both DNA strands but in opposite direction. As you can see, for example, this is one of the DNA strands as the under one is the one of DNA strands. Okay, 5' GAA TTC T'. The down one is 3' CTT AAG 5'. Yes, so this is called opposite direction. So, there is two types of cleave. The first one is sticky end. So, this sticky end, this is the shape of the sticky end. Okay, the second one is blunt end. Blunt end will produce this shape. So, we move toward to example of restriction enzyme. The first one is EcoR1, produced by E. coli. It recognizes the base sequence of 5' GAA TTC 3'. It also makes staggered cuts within the sequence. It produces sticky end, single-stranded ends. So this is restriction enzyme, recognition of sequence, and it will cleave the types of sticky end. So let's move to second one example. SMA1. SMA1 produced by Ceratiria marcescens recognize the base sequence of 5' CCC GG3 3'. The cut is made between the adjacent C and G. Cut straight across the double helix and produce blunt ends. As for example, so there is, and this is the shape of a blunt end. Okay, let's move towards to BAM H1. BAM-H1 produced by Bacillus amyloliquefacins recognize the base sequence of 5' GGA TCC3'. It makes staggered cuts and produce sticky end. So this is an example. So it will produce sticky end.
Assalamualaikum and hi, this is for the third tools for recombinant DNA, which is DNA cloning vector. Okay, DNA cloning vector is DNA molecule which can carry DNA fragment into a whole cell and then replicates. Okay, DNA cloning vector is be able to carry foreign DNA. Then foreign DNA will be inserted into host cell, then replicates into the host cell, which that's for DNA cloning vector. Okay, DNA cloning vector has their own characteristics, which is be able to accept foreign DNA. Second, be able to replicate freely in the host cell, and the third, the last, parser selectable genetic marker. This is for DNA cloning vector. Example for DNA cloning vector, which is we have plasmids for DNA molecule that can carry foreign DNA into a host cell and then replicate. Okay, the capacity to carry foreign is 1 until 10 kb, which is kilo base, just a small one for plasmid. So this is for example for plasmid PUC18. And this is for bacteriophage, example lambda 2001, a virus that infects bacteria, and capacity to carry insertion is 8 to 20. And this is cosmic, example cosmic as COS-1, which is hybrid vector combination of plasmid and cos gene or phage DNA from lambda bacteriophage. Cloning capacity is 35 until 50. And this is the largest one, YAC, which is YAC. The YAC is fragment of DNA from the human genome inserted into a yeast cell. Cloning capacity is the largest one, 100 until 1000 kb. Example for cloning vector.
Hello, my name is Marsha. The fourth tool for the recombinant DNA is host cell. In order to replicate, the cloning vector carrying the target gene, which is the recombinant DNA. The recombinant DNA must be introduced into the host cell. Next point is a host cell is a cell which is able to accept foreign DNA. That is the cloning vector and allows vector to multiply. A host cell can be a prokaryotic cell, such as bacteria and E. coli, or eukaryotic cell, such as animal cell and yeast. The characteristic of host cell is able to receive recombinant DNA through transformation process. Second, able to maintain the structure of recombinant DNA from one generation to another. Third, able to amplify the gene product from the recombinant DNA. Thank you.
Hello, my name is Ariza, and today I want to discuss about the fifth tool of recombination DNA technology, which is modifying enzymes. The enzymes involved are DNA ligase and tag polymerase. Okay, now we are looking towards the DNA ligase function. The first function is to add nucleotides to the 3' ends of primer in order to produce new DNA strand. Second point is by catalyzing the formation of phosphodiester bond to form recombination DNA. Okay, now we are looking towards to the tag polymerase enzymes. Tag polymerase used in polymerase chains (PCR). Second, it can replace the DNA polymerase because of the temperature condition of PCR. Tag polymerase also join the foreign DNA fragment containing gene of interest and bacterial plasmids.
first bacterial cell and human cell. This method, first step is isolation of gene. Plasmid of bacterial cell is isolated. Gene of interest is isolated from DNA donor cell. Okay, this is plasmid isolated. This is AMPR gene, and this is Z gene, and this is bacteria plasmid. This is DNA isolated. This is target gene. This is gene. This is DNA gene.
The next step for method in gene cloning is cleave part of DNA. DNA donor from human and plastic bacteria is cleaved using compatible restriction enzyme. For this method gene cloning, we take E. coli's bacteria as example. So E. coli is produced by E. coli as an enzyme to make a restrictions, and to make as a restriction enzyme we call as restriction sites. The type of the restriction sites is we call as blood end. Okay. So this is model for plasmid isolated and DNA isolated. So example bacteria that we take is E. coli. E. coli will produce E. coli. So E. coli will make a restriction enzyme to cut this part and make this part. The types of cut is blood end. So the DNA isolator also will cut for this part. This part we call as a target gene, which is insulin gene. Insulin gene will insert to plasmid. So that's all for cut process.
So, the third steps in gene cloning is insertion. DNA fragment or gene of interest is inserted into the plasmid, joined together by DNA ligase, forming recombinant DNA.
Process is insertion of DNA that is produced recombinant DNA plasmid. So the next process is transformation and amplification. Transformation of recombinant DNA into bacteria host cell and amplification of recombinant DNA. On this process, the recombinant DNA plasmid inserted into the bacteria through transformation process. Then, as the bacteria reproduce, the recombinant DNA is also replicated, as we know, be cloned. So, this diagram is called as recombinant bacteria.
Recombinant DNA. First, we must place the bacteria in the plate in agar containing antibiotic ampicillin and HCl. This thing is used in the plate. Okay. Now, this thing is want to differentiate between the bacteria that can resistant to the antibiotic ampicillin and the other one not resistant to the antibiotic ampicillin. So the bacteria that will not resistant to the antibiotic ampicillin will not grow, while the other bacteria that resistant to the antibiotic ampicillin will grow and reproduce and form colonies. Okay, the white one, colonies with recombinant plasmid. Okay, then the blue is colonies without recombinant. The white one, recombinant plasmid do not contain intact lactate. Then the white one cannot produce galactosidase. Then the white one also cannot hydrolyze HCl, while the blue one can produce galactosidase and also can hydrolyze HCl.
From us,
Thanks for watching our videos.
a big thanks to our beloved biology lecturer, Madam Ruzia,
Thanks to my teammates.
For your effort to finish this assignment.
Bye! Tabik!
Thank you.